Hello, my name is Kiera McLeod.
My name is Alnudengwa.
And my name is Piara.
And my name is Nathan.
And our project is
This birch is driving my Karaisi.
Our question is, how does the presence of symbiotic fungi and distance from the glacier impact nutrient content in dwarf birch? And we have two hypotheses. The presence of ectomycorrhiza will help birds get more nutrient because the fungi forms a symbiotic relationship with the plants that can provide nutrient and water. And the second is the nutrient content of the birds will decrease as the distance from the glacier increases, because plants near the glacier will benefit from the light reflecting off of it.
So what exactly is birch? Dwarf birch, aka Betula nana, is a low-lying bush, and it usually is found in colder climates. It is important because it is an integral part of the herbivores or plant eaters in Greenland, such as the musk oxen, caribou, and the Arctic hare. So we will be measuring the health in terms of leaf greenness and the number of reproductive organs or catkins of a birch. And you can see that in the figure up above. A is the female catkin and B is the male catkin. And female catkins are require more energy for the plant to make than the male. And dwarf birch is actually one of the plants that forms a relationship with ectomycorrhizae, which you're probably wondering what exactly that is. So mycorrhizae is a symbiotic fungus that works with the host or the dwarf birch, as we discussed earlier, and it exchanges nutrients and water, carbohydrates for nutrients and water. And this is important to the plant because mycorrhizae can have a positive or negative impact on the plant by either giving nutrients and water like we talked about before, or taking too many carbs, more than the plant is able to produce. Ectomycorrhizae are the particular mycorrhizae that we will be looking at, and those are found on the outside of the dwarf birch root as opposed to the inside.
This is where we choose to collect our data. First, we went to Russell Glacier. If you see, it's number 1. We chose Russell Glacier because it's closer to the ice. And for the second location, we chose Lake Ferguson because it's in the middle. And for the third one, we chose Kellyville because it's further from Russell Glacier. In the first picture, that's Russell Glacier, and in the second picture, that's Lake Ferguson, and in the third picture, that's Kellyville. And this is us collecting data. In the first and the second picture, you can see Pia measuring the height and the width of the branch. And in the third picture, we're measuring temperature and the wind. And for the last picture, Sarah is collecting the soil. For the first picture, Sarah is also collecting soil, but it was the better method. And in the second picture, this is us going to another location. And in the third picture, that's Pia collecting the data.
Okay, so when we came back, we had to collect some more data. So we actually have 2 different methods. And day 1, we were measuring the width of each of the plant, the stems of the plant. So we collected 10 stems from one plant, and then we measured 5 leaves from each of those plants. And we were going to calculate that to see the nutrient quality of the plant, but it took a very long time. So we had to come up with another way. So day 2, we realized that it would be more efficient to actually take pictures of the leaves, and we took leaves from all of the stems and put them into these uniform areas. And then we would upload those pictures to a software that analyzed the RGB proportions of the plant, which is red, green, blue. And then the green proportion would tell us the nitrogen content of the plant, which is a great measure to see how healthy a plant is.
And analyzing the roots. We analyzed the roots, at least 5 roots from each soil sample, 195 total. And things we looked for is thin roots, as you can see up in the picture, and the root tips, ectomycorrhiza, fine black hair-like structure. You can see it, this tiny hair-like structure up there, either wrap around or running along the roots.
So then once we collected all that data from in the field and in the lab, we wrote it all down and organized it in a spreadsheet. And from there, we did some statistical analyses on it. So using that data, we found significant differences in traits of birch between the 3 sites. And specifically in 2 of the comparisons, we found significant differences. The first of which was significant differences in height by location. And the second one was significant differences in average leaf greenness. And as you can see, the red on the graph represents Kellyville, the green represents Lake Ferguson, and the blue represents Russell Glacier. And what we noticed was that the site with the tallest plants was Lake Ferguson, which was interesting because it's in the middle of the sites we went to. And then the site with the highest level of average leaf greenness was actually Kellyville. And this was also pretty interesting because we had assumed that the healthiest plants would be both the tallest and have the highest level of leaf greenness. But there were 2 separate sites that had those metrics. So that made the data a little more interesting or skewed to look at.
We also found some other patterns in the data that they didn't have significant differences, but they were still observations that we noticed. So you can see in the bar chart that shows the number of male versus female catkins, and we actually found more female catkins from all 3 sites. And this is interesting because, as I mentioned earlier, it requires more energy for the plant to produce female catkins. So we had assumed that there would be more male catkins. And then on the other chart, you can see it is the relationship of the mycorrhizae to the average leaf greenness. And it actually shows that as the ectomycorrhizae increases, the average leaf greenness of the plants decreases. So this actually went against our hypothesis. And this might possibly be due to the fact that the fungi is taking more carbs and negatively impacting the plant.
Right. So before we go to the conclusion, we just want to remind you all of what the question was. So our question was, how does the presence of symbiotic fungi in distance from the glacier impact nutrient content in dwarf birch? And then again, our hypotheses were, the first one was, the presence of ectomycorrhizae will help birch get more nutrients because the fungi forms a symbiotic relationship with the plant that can provide nutrients with water. So for this hypothesis, our findings, though they were not statistically significant, the pattern that we found actually would go against this hypothesis, as we found that the plants that had a higher ratio of fungus had a lower level of greenness, which was associated with the nutrient content of the plant. And for the second one, our data also went against it because in the location farthest from the glacier, we found higher nutrient content of the plants as well. So actually the plants farthest away had highest nutrient content, whereas initially we believed it would be highest closer. So for our conclusion, we had mixed results overall. Our data did show that the height of the plants and the greenness of the leaves were affected by location. And we did see that higher ratios of fungus in the plant samples were correlated with lower average levels of leaf greenness. However, we couldn't make a decisive conclusion about these results, as they could be affected by many other factors than just the distance from the glacier and the symbiotic fungi. And these factors could be moisture content, elevation, sample size, and many other things associated with time and money that we just didn't have for this experiment.
Questions?
Thank you, ecology group. Are there any questions for the group? You can put them in the chat, or you can also use the raise hand feature on Zoom.
We have a question here, Lauren.
Okay.
When you took photos of the plants for your MPV imaging, was it in the same light, or did you do your best to try and do that?
Can you repeat the question for everybody?
Okay, so the question asked was when we took our pictures of the leaf samples within the grid for our red, blue, green, to try to find the green proportion of the plants, the person who asked the question was wondering if we did it all within the same light. And yeah, we did. We did it within the same room and with the same light source. We tried to keep the pictures consistent in that way.
I have a question. So this year in Kangerlussuaq has been an unusually wet year, and I'm wondering if you thought about soil moisture and how the sort of extreme soil moisture this year may have impacted the results that you found.
Yes, so when we were actually analyzing our data, that was one of the things that we did consider have probably affected our results. And we actually did see that at Lake Ferguson, the plants seemed to grow a lot taller. And we did think that had to do something with the moisture content of the soil compared to Russell Glacier and Kellyville.
Thank you. And there's one question in the chat, and then after this, we'll move on to the next presentation. There's a question in the chat about wind. Was there any difference in the wind?
Yes, so
At Russell Glacier, there was a lot more wind, and at Lake Ferguson, there weren't a lot of wind, and at Kellyville, it was even there was no wind.
Mm-hmm. And also it's hard to associate the wind readings that we found with the actual data that we collected, because also we went on 2 separate days for the collection of our data. So the first day just happened to be a lot windier as well. So that's not necessarily representative of how the sites are on a consistent basis.
Great. Thank you so much, ecology group. There's one more question in the chat, but I think for the sake of time, we will move on and come back to additional questions if we have time at the end. So round of applause for the ecology group. Well done. Okay, and the next group up is the soils group.
we just wanted to give a special thanks for everyone listening, and the government of Greenland, the Ministry of Education in Denmark, the National Science Foundation, Dartmouth College, our mentors and fellows, and especially to Reedy and Sarah that helped make this project possible. Thank you.
Okay, take it away, soils team.
Hello, everybody. My name is Jesper.
I'm Brianna.
I'm Miranda.
And I'm Parth, and our group will be talking about biodiversity and characteristics of the Kangerlussuaq area pertaining to soil. So as you're watching our presentation, we want one question to hover over your mind. Why are these areas so different from one another despite only being 40 kilometers apart? Off of the bat, some things you can see is that on the left, you have a glacier, whereas on the right, you seem to have a mountain with a large pond and lots of greenery, whereas on the left, you don't really see any greenery at all. So we kind of went into our research project with this in mind, and that goes right into our research question.
So the question we investigated during this project was what will impact the ability of the soil in the Kangerlussuaq area to sustain life? And to answer this question, we investigated the chemical properties of the soil as well as microbial life that exists within it and the composition of the soil.
So we had the hypothesis that the soil further from the ice would have unique chemical and physical properties that would support higher biodiversity, so more living organisms, as they'll have been uncovered for longer and thus will have had more time to be acted on by biological and geological processes that support healthier soil.
And to be able to study the soil and the biotope in this area, we have been looking at different features such as moisture, physical composition, nutrients, based on the distance from the ice sheet, which also shows the time uncovered from the ice sheet, the different locations. In biodiversity, we've been studying the growth of soil microbes and the number of plant species. And this map shows us the different sampling locations we've chosen. They are based within a range of 43.4 kilometers. With the 0.660 at the moraine on the far right being the youngest soil, which has been uncovered from the ice sheet for about 50 or 60 years. And on the far left, we have Kellyville, the oldest soil, which is about 7,300 years old and is the oldest testing site.
So here you see the process we use to collect our information. We'd start off going to our different sampling locations and collecting a spot or a plot of soil. We take photos of it and then bring back samples to our lab where we dried them out to collect information on the moisture level as well as burning them, as you can see in the top right corner, to test the amount of organic matter. We also created mud shakes that we use to test pH and other different chemical properties, and you can see Parth also looking at the composition within there.
So the first thing we chose to analyze is soil composition. So soil is a mixture of sand, silt, and clay, with sand having the largest particles and clay having the smallest particles. So this graph, both of these graphs show the distance from the ice sheet ascending from left to right. So on the left, you see 0.660, the moraine, which is the youngest, and on the right, you see Kellyville, which is the oldest. And as you can see, as you get further from the ice sheet, it seems that the concentration of sand decreases and the presence of silt and clay increases.
Yes, and as Path just mentioned, as the soil ages, the particles will change. And this is due to weathering, and this can be through rain, for example. It is mechanical work which will break down the particles within the soil so that the sand percentage, as you can see on this graph, will lower as we get further away from the ice sheet.
We also decided to analyze nitrogen, and we were shocked to find that there was a consistent trend of nitrogen depletion in the soil, except for one sample that we took from Salt Lake.
Another element we tested for in the soil was phosphorus, and we found that as distance increased from the ice sheet, the phosphorus levels in the soil actually increased as well. So there was a positive correlation between the distance from the ice sheet and the phosphorus. And we did actually find the same trend in the potassium levels in the soil as well. The three elements that we ended up testing for were nitrogen, phosphorus, and potassium, which are essential for a lot of plant life and for healthy soil. So as you can see, nitrogen is strongly correlated with the presence of healthy green foliage. And you might not see this in Greenland as much just because there is a lot of nitrogen depletion in the soil. But as the distance from the ice sheet increases, the things that phosphorus and potassium help with, like strong roots and healthy plant growth, are going to increase as well. And we saw that as we increased our distance.
So as was mentioned in our methods section, we also dried and then burned our soil samples. And in doing that, we were trying to see how much organic matter burned off. So organic matter is any decaying material. So for example, dead animals and plants that's within the soil. And when we burn the soil, we can see that amount will burn off. And then the percentage that remains allows us to estimate just how much that decaying life was in the soil. So we found the highest decaying matter content at Kellyville, our oldest site. But as you can see, these results were a little skewed and kind of inconsistent. We have one very high level and then the rest are a little uneven. So there wasn't a strong trend. And then notably, we also found a very high percentage of organic matter in the permafrost. So that's an underground layer of frozen soil that sits throughout the Arctic Circle. And what's important about that is any of the carbon and the other nutrients that are in that organic matter level, if they're in the permafrost, are essentially frozen and removed from the nutrient cycles. So now coming back to our question about supporting life, you might be wondering kind of how we're representing life. So we chose to do a count of the plant species in our different areas and also to culture soil microbes from each area. And we found a slight trend towards higher biodiversity further from the ice with, once again, a significant number of outliers. So it's still not a strong correlation. And we also found, interestingly, that bacterial life was present in all of our samples, including notably those permafrost samples.
So what exactly does the biodiversity data tell us? First, as per plant diversity above the ground, a greater diversity of plants was slightly correlated with more diverse soil compositions. This means that when we saw higher biodiversity, we were able to see more concentrations of silt and clay, as well as more concentration of phosphorus and potassium. Additionally, we saw that plant diversity increases and abundances change as new soils and ecosystems change over time.
So for that microbial diversity, seeing it throughout all of our samples is really important because microbes form the baseline of the food chain, supporting that kind of plant growth we're seeing in our more complex environments, but they can also be some of the first organisms that break down soil and make those healthier, more nutrient-rich soils. Interestingly, we also found that locations that had higher organic or decaying materials generally had higher microbial diversity, suggesting that those soils might be feeding those bacteria better.
So the data we collected ultimately supported our hypothesis, and soil that was farther away from the ice sheet that had been uncovered and exposed to the elements for longer actually supported higher levels of biodiversity. We do want to make note of, however, that because we wanted to take a wide variety of samples, that we weren't able to control for many variables, which is why you kind of see outliers in our data.
Any questions?
Thank you, soil team. There is a question in the chat, which is why does the soil not have nitrogen?
We actually prepared for this answer. So if you go to the last slide, yeah.
that's
Keep
The
going.
last slide, yeah.
One more.
One more.
So as you see, yeah, this is what I researched, but I just wanted to say that as you saw in the graph, there was a consistent trend of nitrogen depletion in the soil. So there are a variety of reasons as to why this happens, and here are some of them. First, low temperatures slow down microbial activity, which essentially reduces the decomposition of organic matter and makes it difficult for nitrogen to cycle within the soil. Additionally, permafrost or frozen soil, as Miranda mentioned earlier, restricts the nitrogen uptake by plants. Third, there are also limited nitrogen inputs to the Arctic soil, and shorter growing seasons also limit for plants to absorb and utilize nitrogen. Also, nitrogen fixing bacteria, which is an essential part of the nitrogen cycle, typically lacks in cold and nutrient poor climates. Lastly, we want to conclude by saying that Arctic soil and the weather conditions here facilitate the ability of nutrients to leach through the soil because of such high sand concentrations, which can result in the loss of any available inputs of nitrogen.
Are there other questions from Greenland, from the Zoom audience? Okay, there's another question in the chat. Is there one coming from Greenland too?
No.
Okay, in the chat we have, could you add nutrients to the soil to be able to raise a garden in a greenhouse type setting?
Our best answer is probably, but those nutrients are going to have to be replenished because they're not naturally occurring. So while you could create a temporary agricultural spot, it's not going to last forever, at least until the Greenlandic soil has been exposed to the elements long enough and has that time to become really fertile for the plants that you would need to grow.
Yeah, I guess I would kind of completely agree with that and say that like we kind of saw from the very broad range of things that we were studying, the generation of more fertile soil or soil that supports more biodiversity is a very complex process because it requires not only those nutrients, but things like microorganisms in the soil that are going to recycle those nutrients and make sure that plants can grow not only like now, but in future generations. So I think that is something that would be a possibility, but I think it's also important to keep in mind the much broader ecological processes that go into having truly healthy soil.
Next slide.
And some final conclusions are that the soil, which has been uncovered for longer, we have been looking at, we can state that it's more complex and diverse. And we can state that the ice sheet retreats at faster rates, then more granitic soil will be uncovered, but it will still need some time to develop the fertile traits we see in the Kellyville sample site, for example. Also, we found the highest amount of organic matter in the permafrost, which leads us to the question, what will happen when the permafrost melts due to climate change?
And we would like to say thank you to our mentors on this project. So our graduate fellow, Fernando, as well as Espen, Nino, and Matt, the other Dartmouth graduate fellows, the JSEP program instructors, the National Science Foundation, Government of Greenland, and Denmark Ministry of Education, and Dartmouth College for this wonderful opportunity.
Thank you, Soils team. Very well done and great answers to the questions. So thank you to those first two groups for presenting. And so we're going to turn from biological systems over to physical systems and next hear from a group who worked on properties of snow.
Is this yours?
Yeah, that one. Studying eyes. Can we move the chairs? Are we allowed to get
That's you, right?
Are we allowed to stand? Yeah. So for the past few days, we have been studying ice at point 660.
Yeah, this is a part of the Joint Science Education Project here in Greenland. And my name is Stine, I'm from Denmark.
My name is Remo and I'm from Denmark.
My name is Mina, I'm from Finland.
My name is Micah and I'm from the United States. So to understand our research project, first you have to understand what is albedo. And albedo is defined as the fraction of light that is reflected by a body or surface. As you can see in the first diagram on the left, there's more sunlight being reflected off of the ice versus being absorbed into the transparent water. Lighter colors would have a higher albedo, while darker colors have a low albedo. For example, on the picture on the right, the dark black asphalt would heat up and be a lot hotter than the grass would on a hot summer day. And that's because it is absorbing more sunlight. And understanding albedo can help predict climate change.
And as you can see in this picture, you can see factors affecting albedo. And in the first one there, you can see the two big rocks, and there are space between them, and that's why they are high albedo, and these rocks are heavy, and that's why it has high mass. And in the second one, you can see these small dots that are small particles in the surface area, and that's why it has a low albedo and low mass. And you can see that from the and how sunlight reflects to the absorb. Yeah.
So the darker the particles are, the lower is the albedo, which is the third one, and the fourth one is when the surface is so clear that it's the background that's reflected. So that could be moraine ice. So the ice on the top is so clear that the moraine under is reflecting the albedo instead of the ice, which makes the albedo lower.
Yes, so we made our research question by debating different questions and figuring out which one would be the most interesting for us to do, and that ended up being how does particle content affect the albedo of ice? And then we made some hypotheses that would make sense, and that would be a higher particle content results in a lower albedo, and then also a higher elevation would result in a higher albedo.
So our data was collected from 6 to 7 different sites on 2 different days. We collected our samples from 0.660, which you can see on the left-hand side. And then in the right-hand side, we also made a map showing where we collected each one of our samples from while we were at 0.660. If you take notice of the black ice, that would be represented by the moraine ice in our data points. And then the clear ice is represented by the clean ice in our data points. And you might notice the 2.5 next to the clean ice, and that is because we took 2 clean ice samples on the second day, because our first day samples, we couldn't plot them because the coordinates got a little messed up.
And where did we find the samples? We found the samples in the different locations, and as you can see in this picture, we have some high elevation and from low elevations, and from clean and dirty ice. Yeah. And you can see them in this picture. And you have some moraine ice in the right corner. Yeah.
So what did we do in the field? Our first step was to clean our shovels and containers to make sure there weren't any extra contaminants in our samples. After that, we identified our test sites, so where we were going to take samples from at 0.660. After that, we used the radiometer we used it face down to measure the reflectance of sunlight being reflected off of the surface. And then we used it face up to measure the amount of sunlight coming in. After that, we took the air and surface temperature with a thermometer. We then collected our samples. And after that, we used a GPS to mark our locations. And then we repeated steps 3 through 6 for each one of our samples.
Then what did we do when we got back to the lab? First, we needed to melt all the ice samples, so it was possible to separate the dirt particles from the water. But before we did that, we scaled each container with the water and particles inside, and then after we filtrated it, we weighed the container by itself, and then we subtracted the container from the weight, so we know how much was in the container. And then we used a vacuum filtration system to filter the water you can play it. So the water goes onto the top, and then the particles will be stopped at a filter, and then the rest of the water will be going through. And then we scaled the particles that were on the filter, And then we use that data to make calculations and also plot data on plots.
So this is our plots. You can see the really high one, the gray at the top. That's the clean ice from day one. And we think that is incorrect because we might took the albedo up twice instead of both down and up. And then we have the two pink ones to the right. And that's the moraine ice. So that's yeah, so the albedo is really low because of the because of yeah, because of the moraine under the ice. Yeah, so we're gonna zoom in on the others.
So now here we have the zoomed in plot of the more stuck together ones in the corner to make a little more sense of them. And there we have the two blue ones, and the two red ones. Those are the high and low elevation ones, and those very much would depend on the weather. So if it had been raining, there would be less particles on the top of the ice, and then the rest of the dirt would fall down to the lower elevation ones, and that is very clear in our data that the lower one is more dirty, and also the higher is higher albedo. And then also we have the green one, which is supposed to be both very dirty, but we have the one to the right where that makes sense because it is both dirty and low albedo, but the other one that was supposed to be dirty seemed to be very clean in the particle amount, and that might be because of the second reason that the particles are very thin layer on top, and then we took more ice than dirt from the top.
Yeah. So the two purple ones and the gray ones, we looked for the same spots but different locations. So we looked for some ice near water with running water, and that's why it might be a lower albedo. It's because of the water has a lower albedo in general than ice, and the clean one is, yeah, so it's the clean ice has a higher albedo, and we looked for clean ice, something that wasn't dirty.
So what can we conclude? Our data likely supports our hypothesis that a higher particle content result in a lower albedo, and a higher elevation result in higher albedo.
Yeah, but we don't have enough time or data to confirm or deny our hypothesis 100%.
What we did do was we tried to make a graph showing the data, and we made a exponential regression going downwards, and then the fact that it is going down is a good sign that we're on the right track, but more samples would make it more precise because the R-squared value is not incorrect. It is not correct, or accurate.
After that, we'd like to say thank you to our fellows, Anna and Elise, as well as Nino for helping us out, and as well as our mentor, Ulak. And we also want to say thank you to our sponsors and everyone else who made this project possible.
All right, thank you, team. So we have a couple of minutes for questions. Please put them in the chat, or feel free to raise your Zoom hand or in-person hand if you are in Zoom rooms.
Can I add a comment first?
Yes.
We were supposed to do all of this with snow, but we weren't able to because of the weather conditions here. It had been raining a lot and the snow was gone on the ice, so we did ice instead.
We have a question in here, so go ahead, Karen.
So what do you think do you think your results would have been different had you used snow instead of ice?
Just repeat the question.
Yes, she asked us if we thought our results would have been different if we did it with snow, and snow has a higher albedo than ice normally does, so we would have expected a higher albedo.
I have a
Yeah.
question.
Yeah.
What was the most surprising thing that you found?
What was the most surprising thing we found?
Snow.
Snow. We
Like
found
one
snow.
sample?
Well,
And maybe the one that particles that where the surface was dirty, and when we dig into the ice and the ice, there's no particles at all on the particles. Yeah, in the ice.
Yeah.
And there's one more question in the chat before we move on, which is climate change impacting the weather there? Is it normal to rain during that time of year?
Lauren, can you repeat? We were clapping. We just get excited here. So
Of course. So in the chat, there's a question, is climate change impacting the weather there? Is it normal to rain during that time of year?
it is not usually that it rains this much, but sometimes rain, but it has been unusually wet this year, I guess.
My name is Leah and I'm from Denmark.
And my name is Nikolai and I'm from Denmark.
My name is Monica. I'm from Finland.
And my name is Max and I'm from Greenland too.
And we are researching ice sheet dynamics with a focus on calving.
Down here.
All the way down in the
It's like frozen.
Let's go. Progress. Wait.
First, we would like to show a video of a calving filmed at Russell Glacier because it's cool. And it's part of the reason why we chose to research on calving. And it was recorded by Monica. Our research question is, how do weather and the river's behavior impact Russell Glacier's calving rate?
We will start showing you the Greenland show from above, from NASA, from outer space. And here you can see Greenland and all its coastal spots. All the spots without the white is the fjords where the ice most likely get out. And the white spot is the ice cap, the ice sheet, which covers 80% of the Greenlandic landmass. What is the Greenlandic ice sheet? This is a cross section of the Greenlandic ice sheet, as you can see, the ice reach up to 3,000 meters. And the middle is the accumulation area where the ice built up with snowfall. So when it snows, the ice will stay neutral. But in the sides, due to gravity, the ice will melt to the sides or have a loss through calving. Yeah.
And what is calving? Calving happens if a piece of ice falling off the glacier, and there's two types of calving that can happen. There's one marine terminating calving and the land terminating calving.
And here's a picture of the principle of calving. It is also a cross section where you can see the flow direction of the glacier. At a certain point, the flow becomes faster, and that leads to the zone of ice stretching and making these crevasses. Combined with the lake that provides a low basal drag, weakens the ice so much that it calves into icebergs. Yeah, and then we want to show you how it looks before and after a calving event, and how we measured calving events with our time-lapse cameras. Yeah, so this first picture and second picture and third picture. And you can see these two big calvings. We could compare the different pictures hour to hour and see when calving events happened.
So our hypotheses were that a higher air temperature will lead to an increased calving rate because a high temperature will weaken the structure of the glacier, causing calving, and that high velocity in river flow will cause an increase in calving rate because an aggressive river, for example, in front of the Russell Glacier, will also affect the structure of the glacier. And then the third hypothesis is that the weather, for example, the rain and the sun, will also create an increase in calving rate. Yes.
And this is how we collect the data. First, we measured the water level near the CT in the Western River, and we took one measurement every hour in 24 hours. And we also put 3 different location temperature loggers, and they collect the data in 2 days. And also we use the pictures from our time-lapse camera, and they took 1 picture every 1 hour. And in that time-lapse camera, we counted how many calvings happens in our research.
Yes, this is a correlation between time of the day and the temperature at Russell Glacier and water level in Watson River at Kangerlussuaq in a 2-day span. So as you see, the blue graph, the blue line, shows the temperature, the average temperature, and can you press again? It has 3 peaks where the temperature was the highest, and they all are highest in the midday.
Yeah, and then we have the orange dots, which represents the water level in the Watson River right down here in Kangerlussuaq. And something we noticed is that the water level is highest in the mornings. And to explain that, we found out that there is a delay between the meltwater from the glaciers to come down here and affect the water level. And that delay is about 8 to 10 hours.
Yeah, and then we made this graph, which represents all of the calvings in a week from the July 2nd to July 9th. And all of the calvings are accumulated over a time period of 24 hours. And if you look at the tendency line in the graph, you will see that there are most calvings during the night and during the late in the evening, which goes against our hypothesis where we thought that there would be most calvings during the middle of the day, because that's where the temperature is the highest. And then we have another graph which shows the calving spread out through the week during the individual days. If you press again. And if you look at the weather forecast, it rained during the 5th of July and the 6th of July. And the rain then increases the calvings, which is shown in the graph. If you press the next. And then the arrows point to the days where there were the most aggressive calvings, the biggest calvings, which we categorize as a 2.5 till 3 calving.
And then we had to ask ourselves, why are we wrong? Because our hypothesis was definitely not right. So we had to go out and research, and then we found a scientific article about diurnal velocity fluctuations in glaciers. And to be precise, the scientific article was about the Helheim Glacier. They said that the glacier velocity peaks about 6.5 hours after the highest solar exposure. We had to compare that to the Russell Glacier, and according to our measurements, this peak happens 10 hours after the highest solar exposure at the Russell Glacier.
And as Nikolai just mentioned, the highest solar exposure was 10 hours, and that makes the ice sheet melt and makes a lot of melting water, which decreases the friction in the ice and increases the calving due to higher glacier velocity, which results in more calving.
Yeah, and then we can conclude that the sun will create meltwater that will seep through the glacier to the bottom of the glacier with a time delay, which will then cause a calving, and that the rain will also seep through with the meltwater in the crevices of the glacier, also creating calvings.
Our second conclusion shows here.
Press again.
Press again.
And again.
That the river's behavior had no significant effects on the river flow. Yes.
Yeah. The end.
Do we have any questions for the calving group?
You mean Team Ice Cube?
Team Ice
Yes.
Cube?
So do you think you would have seen something different if you had measured a different part of the glacier's edge?
Well, the Russell Glacier is one of the most active glaciers here in Greenland. So if we had to look at other glaciers, we had to do it in a much larger time span. And even though we had measurements for a week, it was not good enough. Like, we had to have more to have a more precise result. So if we had to look at other glaciers, it would have taken much more time.
Det var så kanskje kun den eneste, fordi det andre sted ikke. Det kan det være.
Yeah.
Yeah, de smelter.
Yeah.
De smelter bare.
Yeah.
Say it in English, because you're right.
Right? If we want to see more calvings on the other places, it would be hard to find, because the other places, the glacier melts down, not a calving.
You'd see less calving.
Yeah, and we could not see the calvings because the calving happens only if the ice fall off from the glacier. the other places to smelt and like the
Yeah.
So yeah, thank you for great presentations. My name is Lars Port, and I am an associate professor at the University of Greenland, and at JSEP I also represent the government of Greenland. It has been a pleasure for us to share our science with you, and we hope you have learned a lot more about this wonderful area where we have been working for the past 2 weeks. And I would like to express my thank you to all the parents and siblings, families, the National Science Foundation for funding, Dartmouth College, the Government of Greenland also for funding and support of this program, and the Ministry of Education in Denmark, JSEP alumni, remote JSEP students, Polar Field Service, the cook for the wonderful food, Chris for the house, and Per for all the costs. It has been a pleasure for the past 2 weeks. So yeah, I think that's it for tonight. Yeah.